Hello everyone. I'm very excited to have the opportunity to discuss our recent work connecting novel machine learning approaches to analyzing single-cell multiomic data in order to investigate the regulatory effects of acute exercise in human subjects. Single-cell multiome data is an exciting new tool, not only for granting us a finer cell-level resolution of genomic behavior, but with the individual cell-level connection of gene expression and chromatin accessibility data available at our fingertips, we can really dig into connected regulatory behaviors. Indeed, the ability to link shared patterns of change in each cell in the presence of heterogeneity in cell response in both omes provides a platform of thousands of cases to test potential causal regulatory relationships. With the ever-increasing global challenges of heart disease and obesity, it is more essential than ever for an improved understanding of the benefits of exercise and how to tailor exercise regimens to individual needs. Identifying the regulatory circuits that drive exercise response and how these circuits may differ between subjects is critical for personalizing treatment. In this study, we collected vastus lateralis, part of the quadricep or thigh muscle, from 6 subjects, 3 male and 3 female, 4 who underwent a 40-minute acute exercise bout, and 2 circadian control. Samples were collected 50 minutes prior and 3.5 hours post-exercise intervention. Individual nuclei from each sample were isolated and had gene expression measured with single nucleus RNA-seq and chromatin accessibility measured with single nucleus ATAC-seq. Clustering based upon both gene expression and chromatin accessibility, we identified 14 cell types, all of which were present in each sample, including fast, slow, and intermediate fiber types, as well as 2 subpopulations of FAP cells. Cell type composition varied among each sample, with slow and fast fibers the most common, and lume-positive FAP cells with the most common mononuclear cell type. Differential analysis of pre versus post-acute exercise revealed considerable populations of differential genes and peaks for fiber types and lume-positive FAP cells. Approximately half of differentially expressed genes, or DEGs, were shared by fast and slow fiber types, while each fiber type presented unique differential signatures as well. Pathway enrichment analysis identified conserved pathways across the DEGs for all 3 fiber types, including metabolic and cytoskeletal processes. Furthermore, the DEG members of these pathways shared similar proportions of up and down regulated genes following exercise. Intermediate fiber DEGs were uniquely enriched for cell transport pathways, and slow fiber DEGs were uniquely enriched for nucleotide metabolism pathways. Using this data set, showing significant exercise responses in both omes, we will apply 2 distinct machine learning approaches to investigate the gene regulation, Plier and Magical. Plier is an unsupervised tool to identify shared patterns in expression data and connect these patterns with prior knowledge of pathway information. Here we structure the multiomic data as pseudo-bulk signatures by cell type within each sample and search for conserved patterns across both gene expression and promoter region chromatin accessibility. This allows us to look for genes that have a higher likelihood of exercise-induced regulation of expression via chromatin remodeling. Plier identified 12 latent variables, or LVs, each representing a distinct pattern of expression across omes, cell types, and samples. The majority of LVs reflect a cell type or subject-specific expression profile. However, 2 LVs displayed patterns specific to exercise response, LV9 reflecting conserved upregulation across omes, and LV12 reflecting conserved downregulation across omes. The upregulation LV represented a significant increase in expression in all 3 fiber types post-exercise, an increase in accessibility in both fast and slow fiber types. Genes associated with this LV were enriched for cellular respiration and SLC-mediated transport. The downregulation LV represented a significant decrease in expression and accessibility post-exercise in all 3 fiber types. Genes associated with this LV were enriched for the circadian clock, spliceosome, GAB1 signaling, and the mTOR pathway. Magical, multiome accessibility gene integration calling and looping, is a hierarchical Bayesian approach which utilizes the exercise intervention as a system perturbation on which to anchor the construction of regulatory circuits. Each circuit describes a potential instance of altered gene expression following increased presence or activity of a transcription factor, or TF, and accessibility of its chromatin binding site. 2,025 regulatory circuits were identified, 1,500 of which were unique to a single cell type. Fast and slow fibers and lume-positive FAP cells contain the most regulatory circuits. TFs most frequently connected to found regulatory circuits include PKNOX1 and 2, RORC, NR4A1, PPAR delta, and PPAR gamma. TEAD1 is enriched for circuits in all 4 main cell types. TCF7L1 is the most uniquely enriched among slow fiber circuits, and STAT1 is most enriched among LUM-positive FAP cell circuits. We focused on the identified exercise circuits centered at transcription factor PPAR delta, known to regulate cellular metabolism and mediate exercise-induced vascular inflammation reduction. Pre-exercise levels of PPAR delta were relatively consistent across cell types. However, post-exercise saw a marked increase in expression in fast fiber type. The majority of PPAR delta circuit targets were found in fast fiber and were enriched for metabolic pathways, as well as glucose homeostasis, the latter related to the balance of insulin and glucagon, a known function of PPAR delta. PPAR delta gene targets and associated peaks were consistently significantly upregulated in response to exercise relative to circadian controls in all fiber types. Indeed, among all LVs identified by Plyer representing patterns of exercise response across gene expression and chromatin accessibility, the upregulation LV, LV9, produced the strongest affinity with a majority of PPAR delta targets, and 5-fold more than the downregulation LV, LV12. Today we have quickly shown the capabilities of two methods, Plyer and Magical, to harness the increased resolution of single-cell multiome data to investigate the regulatory relationship between gene expression and chromatin accessibility and how that pertains to acute exercise response in humans. We have identified genes with conserved patterns of expression across omes and connected transcription factors to these cross-ome exercise responses. We investigated PPAR delta, identified in our regulatory circuits and known to be a regulator of cell metabolism and exercise response, and found that both PPAR delta and its discovered gene targets and corresponding peaks were all upregulated in response to acute exercise, that its discovered gene targets were enriched for pathways connected to known PPAR delta function. With that, I'd like to thank members of the Silfan, Troyanskaya, Chikina, and Scott Trappy Labs and the New York Genome Center for their work in this study. Thank you all for listening.